Good morning, students. So this is the continuation of the industrial microbiology and also microbial and bioprocess technology subject. So today we are going to have a session on the fermentation kinetics. So this kinetics part, it's a continuation of the kinetics of enzymes you already studied in your biochemistry paper, and those who are not studied and the enzyme kinetics, this may be quite complex. At the same time, you have to learn this one, because without fermentation kinetics there is no upstream processing. So coming to the growth kinetics of microorganisms, the microbial growth dynamics is a subject of numerous fundamental and applied research studies in modern microbiology and biotechnology. So this usually may be very familiar with biotechnologists, because they are doing a lot of things for the upstream processing, and including the applied microbiologists, they are doing the fermentation process using the microorganisms. So this is going to deal with the product formation associated with cell growth, nutrient uptake, respiration, that means mass transfer, oxygen mass transfer, and other metabolic processes. So in this unit, the kinetics will deal with the cell growth, that is biomass formation, and the rate of nutrient uptake by the organisms, and respiration, that is the transfer of oxygen to the microorganisms from the media, and the metabolic processes involved in the kinetics process. So we can go to one by one. The first one is the kinetic parameters in bioprocess. Usually the bioprocess is a very complex study, and especially the bioprocess kinetics is more complex. So the first one is we should know that the basic principles of the growth curve. You already studied about the growth curve, in your UG level and also in the PG level. At the same time, here we are going to deal the growth curve in the kinetics aspects. The rate of increase in the cells, that means the rate of product formation or biomass formation, is dependent on the concentration of the cells present in the reactor. The rate of increase in the cells, that is how much amount of cells going to be increased, that is the biomass production, it is totally dependent on the concentration of the cells present in the reactor. So this can be read maybe or described in the following formula, because this is a basic mathematics one. You already studied the basic mathematics till 10th standard. So this is more than sufficient for this one. So this is dx divided by dt, and directly proportional to the concentration of X. That is X is the concentration of biomass, and in the bioreactor the biomass concentrations are typically expressed in grams per liter. How much amount of grams of the cells in 1 liter of the fermentation media. So this is very important. The dx divided by dt directly proportional to the concentration of X. X is the concentration of biomass. So the next one, so we are going to equate this a constant that is called a specific growth rate. So the specific growth rate is called as mu. So this mu is going to be represented here instead of directly proportional. So we are going to remove the directly proportional, and we are going to add this mu. That's all. That is the only difference between the previous the formula, that is dx divided by dt or dou x divided by dou t. That is directly proportional to the concentration of X. In the place we are going to put a new constant mu. Mu is specific growth rate. The mu is the specific growth rate. The model of microbial growth is referred as exponential growth rate. The growth rate is highly exponential, and that is what is called as exponential growth model or growth rate. So next point in the kinetics is dry weight. Dry weight is nothing but the biomass, and the dry weight is simply the weight of the cells after the water is completely removed. That is either the removal of water by filtration or centrifugation. Both the processes we have to deal with it. So determine the dry weight. The cells must be separated from the fermentation medium, and only the cells to be separated. We should not consider the insoluble components of the media. The media should be completely soluble. There is no trace of any solid particles in the media at the time of inoculation of the cells. And after the growth rate, okay, maybe after the particular the rate formation, the growth rate, and this one, that is the total amount of the biomass formation, okay, has to be calculated by removing all the cells, okay, from the media. So this removal of the cells, okay, from the culture broth or culture filtrate, okay, that can be done, okay, by either centrifugation or otherwise a filtration process. The membrane filtration technique involves, that is, first we can choose the membrane filtration method, and we can go to the second one, the centrifugation. The first one is membrane filtration. The fermentation broth is filtered through the pre-dried, pre-weighed membrane filter. So usually the membrane filter is the 0.2 micron filter or 0.44 micron filter. Usually the PTFE filters are used, okay, PTFE filters are that will not absorb, okay, any aqueous component of the media. So only the cells will be deposited or otherwise is filtered through that PTFE filter. So this PTFE filter is completely the polymer, and that is there are two pore size will be available. One is 0.2 micron and another one is 0.44 micron PTFE filters. The filter is then washed to remove soluble broth the broth components. The soluble broth components means, okay, this has to be washed completely. The filter is then dried and weighed. So that means after the filtration process, you can wash the filter with the biomass by water or otherwise deionized water, okay, to remove the soluble broth components. Only the cells has to be present, including the cells may be sticked with the components of the media, including, for example, okay, if you are using the beef extract or nutrient agar component, okay, that one, the peptone or otherwise tryptone or something, okay, some proteinaceous components. So this may be sticking on to on the surface of the cells. This has to be removed by washing. So this method, the second one is the washing method, and only after washing the filter is to be dried and weighed. So we know the empty filter, okay, that is before filtration. What is the pre-dried and pre-weighed membrane filter? That is the known weight, and so this is the filter with the biomass. If you are minus this value minus, okay, this value is called as the value of the mass of the how many grams of the biomass in per liter. So this is the formula. Just now I said, okay, maybe typically 47 mm, that is 0.44 and 0.45 micron cellulose acetate filter or PTFE filter, no problem. Dry weight is determined using the following equation, that is the weight of the membrane after filtration, that is G, and weight of the in grams, okay, that is weight of the membrane before filtration, that is the difference value, divided by volume of the sample. How much amount of volume of the sample you are going to use it for the filtration process. And so this one is 1000 ml divided by 1 liter. This is the calculation part. So this is a very, very important component, and you should remember, okay, this formula, okay, for your the dry weight or biomass calculation. Second one is the centrifugation technique involves the centrifugation is a technique that will remove the mass content. Okay, for example, if any product, anything, maybe if it is a cell or otherwise any solid particle, okay, that can be deposited by centrifugation force applied on the particular the solid particle. So here we have to consider that, okay, the media should contain only the soluble components, okay, not insoluble components. Only the cell should be insoluble. Okay, the centrifuging the fermentation broth in a pre-dried, pre-weighed centrifuge tube. Preferably you can use a sterile centrifuge tube, okay, and you can use maybe the centrifugal force on the bacterial fermentation broth, okay, that is approximately 15,000 G. Okay, 15,000 G is a value. Okay, you can set it in the refrigerator centrifuge. Just you can do the centrifugation process and sediment it. After the sedimentation process, you can remove the supernatant and wash the pellet and re-centrifuge maybe 2 or 3 times to remove all the soluble components of the media. Okay, like this step. Okay, like we are washing the removable soluble broth components in the filtration. So here in the centrifugal tube, okay, we are going to wash it, okay, by either buffer or otherwise deionized water, sterile water, and the centrifuge tube and the pellet are then dried and weighed. So the completely dried one, the centrifuge tube with the pellet, okay, that means the biomass, to be weighed. This is the formula, like similar, okay, this one, the same one, the component, the weight of the tube after centrifugation process in grams, and weight of the tube before centrifugation in grams, and divided by volume of the sample in ml, so multiplied by 1000 ml divided by 1 liter. So this is the formula. So based on that we can calculate the dry weight. So dry weight can be calculated by two different methods. One is by filtration method, and second one is this one, the centrifugation method. So already explained, okay, this the use of membrane filtration is restricted to the laboratory scale because that is very expensive. The filtration method is very expensive, and the centrifugation is a very cheaper method when compared to the filtration methods and all. Next, coming to the core component of kinetics, that is the Monard model of fermentation kinetics. The Monard model is Monard is a very famous scientist, worked on, okay, the enzyme kinetics process, okay, in the enzymology. The same thing is applied here, the Monard model. So Michelismeter and Monard, okay, this is the Monard model is very, very famous, and we are going to apply the same model, okay, for the biomass, the production also. The biomass growth is dependent on the nutrient availability. Okay, I already told, okay, this one, the biomass growth, the growth of the cells is completely dependent on the availability of the nutrient, the carbon source, nitrogen source, and the oxygen source, and everything. As a concentration of the nutrient becomes a growth limiting substrate, the specific growth rate reduces until growth ceases to the unavailability of that nutrient. So till the availability of the nutrient, the cells will grow, and that will attain a particular saturated level. Okay, this is the one. For example, if you are using the particular substrate level, you cannot say that, okay, all the substrates, okay, can be completely utilized by the microorganisms. There is a limitation for that. Maybe this is the concentration of the substrate. I will zoom, okay, this one. Yes, you can see here. So the x-axis plotted with the substrate concentration, and the y-axis specific growth rate, that is a mu value. The substrate concentration is increasing from, for example, 10 millimolar, 20, 30, 40, 50, 60, 70, 80, 100, okay, likewise. At the same time, the specific growth rate has to increase. It is not like, okay, this directly proportional, and also, okay, this is going to achieve everything is a straight line, not like that. If you increase the substrate concentration for a particular microorganism, you can see there is a steep increase in the specific growth rate, and slowly it will go to attain a saturated point at this substrate concentration. For example, here I will draw a line, and maybe you can know that, okay, this is the point, okay, for so this is the point. At this point of the substrate concentration and the velocity, okay, that is the specific growth rate is constant, attaining a saturated level. So this saturated level will be achieved, and after that, if you are increasing the concentration also, if you increase the substrate concentration also, okay, there is no increase in the specific growth rate. That means, okay, till this level you can maintain the substrate concentration, and beyond that, okay, no need to increase, okay, that one. So that is the indication, okay, by using this one. And maybe, okay, we can see all the components and one by one. Yeah, this is the one explained in the diagram. And in 1942, the Jacques Monard proposed that the following mathematical relationship could be described, okay, to describe the effect of the growth limiting nutrient on specific growth rate. In 1942, that is a period, okay, majority of the scientists, they are working with the enzyme kinetics part, and these three scientists, okay, the Michelismeter and Monard, and all the three, they were working on enzyme kinetics. And the same point, okay, they worked with the enzyme the production also by using the fermentation process. So this mathematical relationship, okay, that can be used to describe the effect of the growth limiting nutrient on the specific growth rate. So that is, the mu is the specific growth rate that is equal to the mu maximum into substrate concentration divided by, and k is the constant, that is the Michelismeter constant, okay, that is k value. So the here, okay, this has to be noted as not Michelismeter, the Monard constant. The Michelismeter constant, okay, that is represented in enzyme kinetics, but here in fermentation kinetics we have to mention about the Monard constant. So Ks is Monard constant plus substrate concentration is, okay, S. So this you have to remember, okay, this is same like the value of the V0 is equal to, and so V max into S divided by Km plus S. That is the value for the derivation of the V max value, and the velocity of initial velocity is equal to, and V max multiplied by S divided by Km plus S. That is the Michelismeter equation. So similarly, the Monard equation, okay, this is a Monard equation, okay, mentioning that the mu, the specific growth rate, that is mu is equal to mu m. Mu m means a mu maximum, okay, that is why it is given here. Mu m is the maximum specific growth rate, okay, that is called as mu m. It is similar to V max, and S is the same, the substrate concentration. Ks is similar to Km, and here we have to mention, okay, this Ks is equal to, okay, this saturation or Monard constant, and S is called as substrate level. And this equation 13, okay, this can be therefore rewritten, okay, this 13 is equation, so 12, this is the 13, because we have the value of this one, and we are going to put the value of mu in the place of mu. We are going to put, okay, this value. We arrived this value, okay, this is the mu value. So we are going to place, okay, the mu value in this equation, that is mu, that is dx by divided by dt is equal to the mu value given there, okay, in the Monard constant level, and multiplied by x. Just you can see here, so dx divided by dt is equal to the mu m into s divided by ks plus s multiplied by x. So this is the value of the mu, the same. So this one we are going to place in the place of mu, okay, that's all. And you can simplify, okay, this one, and further, okay, this will give the final value. And meanwhile, this the maximum specific growth rate is the maximum growth rate achievable when the concentration of the growth limiting nutrient is not limiting. So you have to be very careful that the mu m is nothing but that is the maximum growth rate, okay. This rate is, okay, this one is a particular concentration of the substrate, okay, the maximum growth rate is achieved. Maybe if you know this value, you can calculate the half the mu m. So this half mu m, okay, from the half mu m level, okay, that is corresponding to the value of the substrate, okay, that is called as Ks value. So this is same thing, okay, we are going to calculate the enzyme kinetics, the Km value, by using the Michelismeter graph, okay. This graph is like a Michelismeter graph. In the Michelismeter graph, we are going to plot the values of substrate concentration, and instead of the specific growth rate, we are going to place the V0 velocity. V0 is the velocity, initial velocity. So similarly, okay, this half V max, this is V max, and this is half V max, the corresponding to the value of the substrate, okay, that is called as Km. So that is the way we are calculating the Km value from the Michelismeter graph in enzyme kinetics. But here the same type of the methodology we are going to follow, but this is substrate is same, okay, like enzyme kinetics. Here instead of velocity we have to use the specific growth rate, and this is instead of V max you have to use a mu m, that is the maximum, okay, the mu value, the specific growth rate, and half of the mu m, okay, this one divided by 2, okay, that is this value corresponding to the value of, okay, the Ks is called as this substrate concentration is called as Ks. By this way, okay, we are calculating the Ks value. So the Monard constant is the concentration of the growth limiting nutrient at which the specific growth rate is half the maximum value. That is why I told half the maximum value. So this one is over, and next one is the estimation of Monard model parameter mu m. How we can calculate the mu m. And in typical batch culture, initially we can take only the batch culture, okay, we are not going to take the fed batch or the continuous process. You can watch the previous video. Okay, what is the meaning of batch, and what is the meaning of fed batch, and what is the meaning of continuous fermentation process. Then only you can understand the concept. Okay, this one. In a typical batch culture, the nutrient availability will generally only become limiting towards the end of the fermentation process. For example, in a batch culture, if you are adding the microbial, maybe the inoculum, maybe after the particular period of time, okay, this the nutrients can be completely utilized, and at the time of the end of the fermentation process, and generally the limiting parameter, that means all the nutrient availability is limited, that means that is going to be decreased. This is reflected in the slowing the growth rate as cells entering into the stationary phase. That is why the growth curve is also, okay, it is completely depends on the nutrient availability. You can see here, this is the growth curve, and this is the lag phase and exponential phase, and this is stationary phase and death phase. And everything depends on the substrate concentration. You can see here, okay, this is like the Michelismeter graph, and here, okay, this is the saturated level, and after that, okay, this is going to be in the stationary phase and going to die, okay, that is a death phase. The analysis of the model, okay, the Monard model shows that, okay, when S is more than the value of the Ks, okay, then the mu this approximately is equal to the mu m. That is the mu m is the maximum, okay, this the levels, okay, that means you can see here, this is the mu m level. So this we are going to calculate the mu m. How we can calculate what is mu m? So mu m is nothing but the maximum specific growth rate. How we can achieve mu m? So this mu m is calculated the saturation point corresponding to the specific growth rate, okay, that is called as mu m. This can be calculated, okay, by using this one. If the mu m is equal to or approximately equal to mu, okay, that means if the substrate concentration is that's equal or more than, okay, that of K5, okay, that is a Ks, okay, that is in that condition the mu is approximately equal to mu m. So I think, okay, you hope understand the concept. For example, here the substrate concentration, I'll explain, okay, again to you by using, okay, this graph. You can see here this Ks. The Ks, if you are using this concentration, okay, and above the Ks value, if you are using it, that is equal to, okay, this one. For example, so this is the value. For example, this is 10 millimolar, 20 millimolar, 30, 40, 50, 60, 70, 80, 90. So this is approximately 90. If you are using, okay, more than this Ks, for example, instead of 20, if you are taking, okay, this 90, so that is equal to Km. That is a micromolar concentration of, okay, this mu m value. That is a specific maximum specific growth rate. So that is why, okay, this we are going to arrive the value of the mu m, that is equal to mu. So during this exponential growth, okay, the mu minus mu m, okay, that is mu is equal to mu m, okay, it is not minus, okay, this is mu is equal to mu m, and based on that, the dx divided by dt is equal to mu m multiplied by x. So here this is not minus, students, and you have to be very careful, and I'll write, okay, this one, I'll correct, okay, this one. So this is equal. So here in this case, so mu and mu m are approximately equal, at which stage, if you are taking the substrate concentration that is more than the value of Ks, that means in this, the graph you can see here, this is the value of Ks. If you are taking the concentration, okay, this concentration approximately, so if you are taking the substrate concentration of this one, so that is equal, approximately equal to the concentration of mu m. So that is why, okay, it is mentioned here. If the S is more than the Km, that this Ks means approximately the mu is equal to mu m. So the mu is equal to mu m means, okay, the dx by dt divided by is equal to the mu into x is the formula. That is, this is the formula, mu into x is the formula. So this formula we are going to put, instead of mu, we are going to put the value, the mu em, that is maximum, okay, and we are going to add this value here in the place of mu we are going to add the mu m. Okay, that is only difference. Okay, this is the formula, and this formula, the maximum specific growth rate can be determined by arriving okay this value. And next, coming to the Monard constant, we already explained okay that about so what is Monard constant? Okay, the Monard constant is nothing but the half the value of the specific growth rate, the interpolated to the value of substrate concentration. Okay, this value is called as Ks, and this is explained okay previously. And next one is the properties of the Ks. Using the batch culture okay to determine the Ks generally leads to unsatisfactory results. For example, okay, if you are using the batch culture to determine the Ks, generally leads to unsatisfactory results, because batch is a controlled one, that is a limited one. Okay, and if you want to get the accurate results of the fermentation kinetics, okay, you have to use the continuous culture media. And this is the one you have to make a note of it. The continuous culture data is required to get the values of the Michelismeter kinetics data, okay, like the alteration and everything, the Ks value. So this Ks value, if you are using the batch culture, that is not exactly correct one. You have to use the continuous fermentation media. Okay, I'll remove all the ink in the slide and take the laser pointer. There is an alteration okay for the Ks also. For example, to determine the value of Ks, we have an alternative use, that is Lineweaver Burke plot and Eadie-Hofstee plot of the Michelismeter kinetics analysis of enzymes. The same thing we can use it, but we are not going to apply here. So this is more than enough for you. And next one is the yield coefficient. Okay, this is called as yield coefficient. Yield means okay how much amount of the products formed. Okay, that is called as yield. So there are two different types of yield which biotechnologists are most commonly interested in. Okay, which one is the biomass yield and the product yield. The biomass is different and product is different. For example, if your product is a biomass, okay, that maybe biomass is equal to product. If your biomass is not a product, maybe if your product is an enzyme secreted by the biomass, the product is different and biomass is different. That is why. So you have to mention okay both in different parameters, that is the biomass yield is different and the product yield is different. For example, the biomass is nothing but a cell and the product is your product. For example, enzyme or protein or vitamins, okay, or antibiotics, whatever maybe you are going to be focused, okay, that is called as product yield. So here y-axis, okay, that is mentioned as the biomass yield, and the product yield is mentioned as the PS. Okay, the P is the product and X is the biomass. The same thing, but Y is the yield coefficient. Y-axis and Y-PS is the product yield. The biomass yield coefficient is the average weight of the biomass produced per weight of the substrate utilized for a batch culture. That is, the Y-axis is calculated. Okay, what is Y-axis? The biomass yield is calculated based on this formula: x1 minus x0 divided by s0 minus s1. That means x1 is the biomass produced after the fermentation process and minus before the fermentation process the value, and the substrate concentration is initial substrate concentration and minus the end substrate concentration. So this one is the formula is very important okay for the enzyme the enzyme kinetics and also the yield coefficient related to the fermentation kinetics. Y-axis is equal to x1 minus x0 divided by s0 minus s1. So next one is x0 and s0 are the initial concentration of biomass or substrate concentrations. You already told okay just now. And next one is the product yield. Yield coefficient means the YPS. Okay, yield coefficient, product yield coefficient. The previously we studied about this is biomass yield. So y-axis means biomass yield and YPS means okay this product yield. The product yield, okay, the after the fermentation product and before the fermentation product, okay, this value, and divided by substrate concentration, initial substrate concentration and end substrate concentration, and both are calculated in a proper way. And the representation of the biomass production, entire biomass production. So note sometimes the product yields are represented with respect to the biomass production. The product yield and also the biomass yield, both are same, means okay, you have to mention okay, this ypx, okay, not yps, ypx, okay, this is called as the relationship between the product and the biomass. So both are we are going to calculate. The P is the product and X is the biomass. And the P1 minus P0 divided by X1 minus X0, okay, that's called as okay, this the yield coefficient, okay, this ypx related to the product and also biomass. So this is separate. You should remember that this is only the biomass xs and only the product ps, and both the product and the biomass, that is ypx. So that is a different, and later yield expressions are sometimes used in the quantifying the production of secondary metabolites, so this may be very useful. So with this I'll conclude the fermentation kinetics, and this one is very important for the section B and sometimes in the section C also. Okay, all the very best. Thank you.